Hey guys, this is going to be a quick introduction to the process of getting from a sequence of nucleotides or genes ultimately up to the production of a protein that can influence genotype. And so we know that there's an inherent connection from all of our investigations in genetics that DNA ultimately codes for the way that we look or the way that any organism looks or functions in some capacity or another. So we need to talk about this process of how we get from a sequence of A's, C's, T's, and G's to the huge diversity that exists within the biosphere. So the nice part is that there are some overlaps with DNA replication in terms of process. That's an efficiency model in terms of evolutionary significance, but ultimately we will move into a little bit more complicated processes as we move into protein production. So let's start all the way back with DNA. So we know that we have this double helix. Let's got a little messy there, but it is what it is. Two-stranded, made up of nucleotides. The sugar is deoxyribose. So there's there's a lot of characteristics we know about DNA. And DNA is the template. So once again, we'll be using DNA as a template strand. Some of the players that will roll over again, topoisomerase, helicase, and then ultimately a polymerase in some capacity, but it won't be DNA polymerase this time because we're not making DNA. So the overall thing that we need to do is we need to get from DNA over to this new molecule called RNA. And specifically, we'll start talking about mRNA, but we'll get there in a second. All right, so what's the difference between DNA and RNA? The major difference is that structurally, RNA is single-stranded, so it's not going to look like a ladder at all. The other unique portion of this is that RNA uses a different sugar, ribose, where DNA uses deoxyribose. So it still has the same kind of structural characteristics. It's just got a couple different hydrogens, a couple different oxygens. But what you need to know is that it's just a different sugar. There are still nitrogen bases in terms of the structure of RNA, which is still kind of nucleotide based. But the biggest difference is there's no thymine anymore. You're using a new nitrogen base called uracil. So when we look at DNA, we know that DNA has A's, C's, T's, and G's. But in regard to RNA, we have A, C, U, and G's. So the nice part about this is that U realistically just supplements T. It is opposite of A, so that when you have an A, you have a U. So it's not really too big. You just have to remember when to use it and when not to use it. So what this first step is taking the template strand of DNA and making this kind of transitory molecule called mRNA or messenger RNA. Now the nature of this is to take the information in DNA, turn it into a sequence that can then be used to make protein. So this process is going to be called transcription. Now this transcription, definition-wise, is the process of taking DNA as a template strand and making an RNA complement to it. And like I said, the big differences are U instead of T. All right, so once you have this mRNA, you have your sequence of nucleotides that will be used to make proteins. So that's where we're going to step into a different process without going too specific. And this is where we're going to take mRNA and we're going to make a protein. Now, a protein is made up of amino acids in sequence. So amino acid plus amino acid plus amino acid, and then so on and so forth. So the nature of a protein, or what it does, is based on the sequence of amino acids. So let's say that we take this mRNA, and I forgot to draw it single-stranded, so it kind of just looks like a squiggle here. And we are going to use that mRNA as a template. So it eventually gets flattened out and then has a sequence of A, U, G, C, A, U, C, C. Notice there's no T's. And you use this as a, let's add another G there, as a template for adding amino acids. Now, you might be saying, we're making a big jump here from mRNA to amino acids. So what's the link here? So we are using DNA to make a template strand, and that template strand will determine the sequence in which amino acids are added together. Now, do you remember that crazy chart on the pretest that had a whole bunch of combinations of letters and then correlated to a different set of letters? What those were triplet sequences that coded for specific amino acids. So whenever we start looking at mRNA in terms of making a protein, we can separate it into triplets. And these triplets will be used to determine which amino acid is attached, because we've got all these amino acids floating around that are just waiting to be attached. And they have these things which we'll find out to be called anticodons. These anticodons will match up with these things called codons. So let's say that this is a U, an A, and a C. So what would happen is, based on this mRNA template, and we can change that, make sure it's labeled right. This is mRNA template. We know that these amino acids, this anticodon will then end up binding with this codon, and eventually we know that AA, number 1, amino acid number 1 would be first. Now there's other amino acids hanging out here, number 2, with an anticodon in GUA, and this guy would then bond with this one. So we then know that amino acid number 2 would be next, and eventually those two amino acids bind together and start to form a protein. And this is the process that we're really looking for. We're looking to use this template to order the sequence in which amino acids are being added, because amino acid sequence ultimately dictates what that protein does, what it activates, and realistically what the cellular or organismal response would be associated with the production of that protein. So there's a lot of other details that I didn't really go over here, but overall, here's really what I need you to understand. Is 1, DNA is still the template, and there's a sequence of nucleotides, and those sequence of nucleotides matter. The process of transcription uses the DNA template to create an mRNA strand. This mRNA strand is the instructions for building a protein, and that process is called transcription. The next thing is, by using that mRNA sequence, you can it will tell which amino acids to bind first, second, third, ultimately creating your protein that does something. This process is called translation. So, things that, the big things is, we will work through the process of understanding all the details of transcription, and then we will work through the process of translation, and then look at using the table that's got the codon and amino acid table, and so that you could realistically go all the way from your original DNA sequence to the appropriate sequence of amino acids. And this is important when we look at mutations. We'll be able to infer how drastic is the effect of a mutation based upon the amino acid sequence, whether it's changed, whether it's not changed, if it is changed, how big is change, how big is the change. And from there, we can talk about physiological implications of modifications that are made at the genetic level.